Hi, I'm Steve Palumbi, and this is Lecture 8 in The Science of the Extreme Life of the Sea. And this is one of two lectures that explores the unique habitats, the deepest places in the ocean, the deepest places that life lives on the planet. On the average ocean depth is almost 4,000 meters. And there's a lot of area down there that organisms can live in. The deep sea area itself is about 160 million square kilometers. And so that makes it the biggest habitat on Earth, three times the amount of forest on land. And it's a very peculiar kind of habitat. It's characterized by pretty low temperatures, between 1 and 4 degrees centigrade, pretty constant in most of the deep sea, um, and a fairly low food supply and pretty high pressure. Well, there are other places in the ocean that are cold. There are other places that have low food supply. And so the one completely unique feature of the deep ocean as a place to live is the large pressure that's there. So this first part, we're going to talk about pressure effects, how pressure changes the way organisms have to live their lives and the adaptations that deep sea organisms have to have in order to basically deal with that kind of pressure. So, what do we mean by high pressure? At 200 meters deep, the pressure of the water sitting above you, pushing down on you, if you were 200 meters deep, would be 21 atmospheres worth of pressure, 21 times the pressure that we see at sea level. Right above the water. 3,000 meters in the deep sea, that pressure is 300 atmosphere, a very strong, relentless push that actually pushes on everything in the deep sea. We can get a little bit of an idea of some of the changes that are happening in the deep sea organisms by the ideal gas law, where pressure times volume is equal to the number of moles in a particular gas, the universal gas constant, and the temperature. So if we know the the numbers on the right side of the equation, we can relate pressure and volume to one another on the left side of the equation. And in particular, that matters for all organisms that have gas spaces in their bodies. And of the marine organisms that we're familiar with, probably the most significant gas space in organisms is the swim bladder of fish. This is sort of a blow up of the guts of a trout. And what you can see running along on top of the gut here is a long, essentially, balloon of the swim bladder. By inflating that, making it bigger, by putting gas into it or deflating it, then the fish can adjust its buoyancy. Otherwise, the fish, which is negatively buoyant, it would sink to the bottom. But fish can maintain neutral buoyancy by changing the amount of gas in the swim bladder and then essentially being able to then adjust that buoyancy no matter where they are. So, kick in the ideal gas law. As this fish were to go deeper and deeper, then the swim bladder would become less and less and less large. It would basically be crushed by the pressure. In order to maintain the same buoyancy, then that fish has to add more and more gas to this swim bladder in order then to be able to stay neutrally buoyant deeper in the ocean. The ideal gas law tells us exactly how much that has to happen. So basically, it happens the reverse if you're coming from the deep up to the surface too. So if a fish comes from one particular depth, say you reduce the pressure by half by rising up, well, then that actually doubles the bladder size. So an example of that would be from 30 feet deep, which is about 2 atmospheres of pressure, to the surface, which is 1 atmosphere. Every fish that makes it from 30 feet deep to the surface, their swim bladder is going to double in size by the time it gets to the surface. Now, fish can deal with that by reducing the amount of gas in the swim bladder and then pulling the gas back out, reducing its size. And that's an active process as fish go up and down in the water column, maintaining their buoyancy by maintaining the right amount of gas in the swim bladder. That's similar to how divers adjust their buoyancy, although fish do it much more elegantly. The buoyancy compensating vest of a scuba diver is used to keep that diver neutrally buoyant. And any of you who have been scuba diving know that as you go down and down and down, your BC vest deflates. Not that there's any less air in it as before, it's just that the pressure goes up and that the volume goes down. And if you want to maintain the same neutral buoyancy you had, say, at 10 feet deep, you have to have a little bit more air in that BC as you go down. But then as you go back up, you've got to release that pressure because as you go up, that same amount of the molecules of air will become more and more and more buoyant. You don't want to go shooting to the surface. And so divers adjust their buoyancy compensator by pushing air in from the tank or releasing it. Same thing with a fish swim bladder. It's just being done metabolically. You can calculate what the necessity is for the buoyancy compensation by knowing the weight of the fish, by knowing the buoyancy of the swim bladder and calculating what needs to be put in. And generally small changes are sufficient for most of the movement of most fish. However, that isn't the case when people go fishing. This is a pretty common view of a fish that's been brought from deep in the ocean, say 300 feet, a couple hundred meters, very quickly up to a fishing boat. What's popping out of its mouth there is its swim bladder. That swim bladder hasn't had time to adjust itself as the pressure has changed, as the fish has been brought to the surface. And so it's way more inflated with gas than it should be. It's so inflated that it comes popping out the mouth of this poor fish. Other kinds of fish in the sea don't actually have the same problem because there's not that many other gas spaces in marine organisms besides the swim bladder. Sharks, for example, don't have swim bladders. They rely on two different kinds of mechanisms to remain up above the bottom. One of them is by actively swimming, and by angling their fins, they're basically getting a certain amount of lift just from swimming ahead, swimming forward. So their sharks are almost constantly swimming in order to stay above the bottom. However, they also are not quite as negatively buoyant as they would be otherwise because their liver is full of very lightweight floating oil, and that gives them a buoyancy that's a little bit better than they would have otherwise. It's also why their livers, being full of oil, have been long sought for their oil content. So sharks, unlike fish, if they're not swimming, will tend to sink to the bottom. They can't hover neutrally buoyant like bony fish can. Well, buoyancy as one area of the effects of pressure also affects all things in a body. And so the next section is about pressure effects, not on swim bladders or small gas spaces, but on the molecules that all marine species have when they're living in the deep sea. Pressure affects all molecules in all organisms in the deep sea, and understanding that tells us something about the adaptations they need to have in order to live there. What I want to focus is on one kind of molecule, lipids, because lipids make up the bilayer that is part of cell membranes, and the state of those lipids and the pressure effects are a really important part of how those bilayers, those cell membranes function in organisms in the deep sea. And to think about that, here's a diagram of two different ways that the lipids in a cell layer can be interacting. One is in a fluid phase where the lipids are there, they're organized in a bilayer, but they're not oriented with one another very well. They're moving around a great deal. Essentially, there's a lot more movement and flow in that part of the cell membrane. On this side, cell membranes are in their sort of more normal gel phase. There's still some movement, but the lipid parts are oriented well with one another, and there's a structure to how the lipids are sitting within the membrane. So you can change from the gel phase to the fluid phase just by heating things up. A little bit more heat will cause them to become more fluid. They'll melt, essentially. But you can also do that by changing the pressure. If you have high pressure, it's easier for the gels to stay in their gel stage. If you have low pressure, then the whole bilayer can shift to its more fluid phase. So temperature and pressure depends or determines the state of these lipids. All lipids are not alike, and some lipids form gels easier than others. And we can talk about the two lipids in terms of how saturated they are. The top here is what's called a saturated fatty acid or a saturated lipid, and that means that of the carbons here, the Cs, all of them have hydrogens. All the possible bonds to this carbon have hydrogens on them, with the exception of this last one here that has a couple of oxygens on it. Now, that's a saturated fatty acid because the carbons are saturated with hydrogens. An unsaturated fatty acid is shown here, where these two carbons have a double bond between them, taking up the space of a couple of the hydrogens that they would have had. And instead of hydrogens being here and here, there's the double bond. So this set of carbons in this fatty acid are unsaturated. They're not completely saturated with hydrogens. There's this a couple of double bonds. Well, the double bond creates a little kink in this structure, and that means that the way these lipids work together inside a bilayer is different if they're saturated and straight or unsaturated and have these little kinks. So this is a diagram of that where saturated lipids only, all of the lipid tails are straight. They form nice patterns with respect to one another, whereas if there's unsaturated lipids in the lipid bilayer, then it adds a little bit more wobble to them. They're not all sitting straight. This structure is easier to melt than that structure. This structure is less easy to make into a gel than that one. So the amount of saturated or unsaturated fats in a lipid then can be tuned depending upon the external temperature or pressure to allow the membrane to have the right amount of fluidity, the right amount of gel. If we think about that, then we might then suspect that different membranes at different parts of the ocean, organisms that live in different parts of the ocean, might be organized differently, and we would be right. We can then actually ask, well, does membrane function change depending upon the external environment that those membranes are tested in? And one good way of doing that is by looking at nerve transmission in fish that are from different habitats. So graphed here is a normal nerve impulse in an Atlantic cod. And these animals are generally living near the surface or down a couple hundred meters. They're adapted to relatively low pressures, and their membranes have the right amount of saturated fats in them to allow them to have the right fluidity at the normal temperature that they're living in. So this is a normal nerve impulse from Atlantic cod. Well, if you take that same preparation in the lab and then exert on it and the same kind of pressure that that animal would see at 3,000 meters, then you get a very different kind of nerve pattern that results. Instead of the normal one, essentially this is squashed. Effectively, the membrane isn't working at high pressure the way it should be at low pressure. Let's suppose we do the opposite. We take a fish that normally lives at deep water, like this lizardfish, look at its nerve function at 3,000 meters pressure, and it's totally normal. If we take it and bring it up to the surface, its nerve function declines a little bit. Not quite so much as the decline of cod at deep, but there is a shift in the nerve function of lizardfish in shallow water. Shows that the lipids here, this fish at deep water, are adapted to be able to be fluid and have normal nerve transmission at high pressure. And it's less likely to be, or less able to be fully functional at low pressures. And that's because the amount of saturated fat in the lipids of different species is in fact different depending upon where they're from. This is just a diagram of the saturated and the monounsaturated and the polyunsaturated fats in different sorts of species. And you can see they're widely variable. Saturated, if you remember, is straight molecules, lots of hydrogens all around. Non-saturated, unsaturated fats means that they have a kink in them. Well, the kinked ones are the ones that are harder to form into gels. So deep sea fish have more unsaturated fats in them. They have a lower level of saturated fats. So this is a diagram of the kind of saturated fat levels that different species of fish have. These are from different habitats. If we just look at the level of saturated versus unsaturated lipids in different fish as a function of the normal depth that they live in, you can see that deep water fish down here 4 kilometers or so have much more unsaturated fats than saturated fats compared to the species that live in shallow water. So the type of biophysics that we would predict from the way the molecules look and the pressure effects is in fact what we see in the species that live in deep and shallow habitat. Other parts of the ocean, of course, are dark, some places and sometimes, but the deep sea is uniformly dark all the time. Well, but it isn't really. Actually, there's no light from the sun that deep, but there is light in the deep sea. And that light comes from the organisms themselves, made by them, and that's called bioluminescence. Well, we can sort of see that in all kinds of critters. This particular animal is an anglerfish. All you're seeing is the bioluminescence of that animal as it's swimming through the dark. And the patterns of that light in those animals is generated by a very specific kind of chemical reaction that has a lot of different consequences. So light in the dark is essentially made by an enzyme called luciferase, and it uses luciferin, which comes in lots of different types and lots of different specific chemicals, combines that with oxygen that produces an activated luciferin complex and light. Now that luciferin complex will then get reconverted back to luciferin, and so this whole cycle can continue to happen, to continue to occur with luciferase specifically causing the light reaction. Well, there's a lot of different kinds of luciferins that are out there. This shows just a different set of them and the kinds of organisms that have that type. And we see different luciferins in lots of different kinds of bacteria, but we also see them produced by animals themselves in the deep sea. A lot of the light that we see comes from bacteria. The bacteria have a particularly active kind of bioluminescence, and it's tied to their metabolism. Now, the luciferase reaction itself takes oxygen, converts it to water, and some people have thought perhaps that was one of the original pathways by which oxygen was pulled out of the bacterial cells because it's a toxin. So when photosynthetic bacteria first evolved a couple of billion years ago. Oxygen was a toxin in these habitats. Could the luciferase actually have been one of the first detoxifying roles of any enzyme in the deep sea? At that point, there were no eyeballs. There was nothing to see light, and so the role of luciferase in early bacterial evolution is a little bit murky. Well, fish can produce light, but they also can house bacteria that are bioluminescent. And this combination of using bioluminescent bacteria to produce light and producing it themselves is one of the complexities of how bioluminescence happen. There's a couple of different features of the way that organisms control light production. So bacteria producing light all the time. Certain crustaceans, for example, control light production by essentially having the luciferin, that is the fuel, and the luciferase, which is the enzyme producing the light, in different glands. And when they want to produce light, then they'll basically mix those two things together and spew it out, producing the light outside. So this is an image of an ostracod that uses light the way fireflies do to attract mates. And I'll show you a few pictures of that later. And what this animal is doing is essentially just squirting out the two components of its light production machinery, the luciferin and the luciferase, to make light outside its body, essentially outside here in the water column. The image I just showed you of that ostracod essentially creating light, that was blue light. Blue and green light. And that's what you mostly see in bioluminescence, like in this picture of coastal bioluminescence in the Maldives. Most of the light produced in bioluminescent organisms, in fact, runs in the blue and the green range. Whether or not you're talking about the bioluminescence of ctenophores, starfish, worms, crustaceans, most of it falls in this particular blue to green range. The reason for that is that that kind of wavelength is the wavelength of light that travels fast or furthest in the oceans, and it allows the light itself, whatever role it's playing, to play that role over a bigger zone. There's a wide variety of different kinds of things that organisms use light for, from startling predators to signaling mates, and they do it in a wide variety of different sorts of ways. For example, a deep sea shrimp, by spitting out the luciferin and the luciferase in different streams outside, can create a luminescent cloud outside its own body and can create the cloud and squirt away, hopefully distracting a predator and attracting it to the cloud of luminescence rather than the dark shrimp that is moving away as quickly as possible. Other organisms do exactly the opposite, and they lure prey with bioluminescence. This is probably the most common and well recognized bioluminescent trick in the deep sea. Anglerfish using their ESCA, this is called, the bioluminescent lure, brings prey in close to their mouth. They're sit-and-wait predators, and as the prey comes in, they can attack them and use them. Other organisms like ostracods use light like fireflies do. They use light to signal their presence, in this case in shallow water environments, mostly in coral reefs. And what's shown in this diagram are the light patterns of different species of ostracods. Those light patterns are thought to be signals to mates. They're different from species to species, even in the same area, and attract the right mate to the right light pattern. The ESCA itself in anglerfish is actually a production of not just the fish, which produces this long dangling digit at the front fin of the front spine of its dorsal fin, but also the light in it is not actually the fish producing it, but is bacteria that live within that organ and produce the light that is then attractive to prey. Countershading is often seen in not deep sea animals, but in animals that are living in the twilight zone between the lighted surface currents and the very deep sea. If you actually are in that sort of zone, then there is a little bit of light filtering down from up above. Countershading is where the bioluminescence of an organism is clustered on the bottom side, the ventral side, allowing the light to essentially be directed downward. This image here shows the silhouette, in this case of a squid, with dim light from the surface showing some predator below, exactly where that squid is. But the counter shading that the squid can have essentially makes up for that small amount of light coming up from above, obscuring the shadow of this squid from any potential predator that is living underneath it, or also hiding it from its potential prey, so it can come in from up above. That's what this pattern of luminescence might look like for a squid from up above. Lately, actually, just in the last couple of days, Ben Burford, who's a graduate student at the Hopkins Marine Station, has just published a paper with a different kind of bioluminescent pattern in squid. In this case, the luminescent pattern itself is a background, and the squid then changes its color against that background in order to signal to conspecifics, other Humboldt squid in the area, and communicate with them. In very dim light, visual communication is practically impossible. And so Ben's proposal is that the animal lights up its body in a low way, but light it up enough so that it can then play its normal games with its other color patterns to essentially communicate with other Humboldt squid in the area. So if you actually add up all the different kinds of roles that bioluminescence plays, some of them are defensive, some of them are offensive, some of them are attractive, then you see that the bioluminescence is a really important part of the ecology and the interaction not only within species but between species, all in deep sea areas. Organisms use bioluminescence, but by using it, they're also at risk because of it. Say, for example, that you want to use bioluminescence to light up the area and find your prey. Well, the light that you use, the bioluminescent light, is incredibly attractive to your predators as well. So there's a danger in using light just as well as there's an advantage to using light. And what we can see in the evolution of a couple of different specific types of fish is a really interesting and clever solution to that problem of using light but not having it be a huge problem for you. So most bioluminescence is in a very narrow range of the visible spectrum. It's the blue-green spectrum. And that's because that's the spectrum or the type of light that travels furthest in the ocean. But that traveling far might not be a huge advantage depending upon what you want to use your light for. The flip side as well is that most of the organisms that live in the deep sea are able to see light, but they're mostly concentrating their visual capacity in that narrow range of bioluminescence in the blue and the green part of the wavelength. Very few of those organisms can see well outside that range. But there is an exception, and those are called the loose jaws. The loose jaws are in a particular family of deep sea fish. They're called loose jaws because they can pop their lower jaw loose, and as a consequence, they can actually eat and attack fish as big or a little bigger than they are. As active predators moving through a big environment without much food and without much prey, they actually have searchlights that they use in order to find their prey. And the searchlight here, you can see the lower end of this eye. It's a patch of bacteria that is putting out light, bioluminescent light. And in this particular case, though, the light that it's putting out is not the same as usual. The light coming out of these particular patches under the eye is in fact shifted much more towards the orange and the red. By then using orange and red light, then these fish can use the light to find their own prey, but very few other fish in the environment can see red light. Their predators essentially then can't see that they are using light to find their prey. Actually, there's another component of this that has to happen, and that's that using red light as a searchlight sort of option is great, keeps your predators from seeing you, but then you need to be able to see that. So these fish have these dual adaptations. They're producing red light, but they've evolved the light gathering proteins in their eyes to tune those to be able to react to red light very specifically. We can look at a couple of different features of that. One is the evolution of these fish and what fish have this ability. This is the phylogenetic tree of a particular type of loose jaw that lives in the deep sea. These are all different forms of deep sea loose jaw fish, and what's shown with the red stars are the fish that have the red shifted visual pigments that allow them to see red light. We can look at that, and from the standpoint of a different phylogenetic tree, these are the protein sequences of the opsins that are used in the eyes to capture light. Now, those opsins capture light very differently depending upon their amino acid sequence. Some opsins capture green light, some red, some blue. In our eyes, for example, most eyes of most color able creatures have a number of different opsins that capture light in different color ranges. In this case, this is the evolution of the opsin genes in a group of species that can and cannot see red light. The ones that can see red light are shown in red here, and then these denote the amino acid changes in the different parts of the opsin gene that tune the protein to be able to absorb light at different wavelengths. In this particular case, this entire clade of species has evolved its opsin genes in order to see red light. There has been a couple of cases of a reversion of that. This group, this other clade within the red seeing clade, has essentially devolved its opsins, changed its opsin back to seeing more normal blue-green light, and you can see the various amino acid changes listed here on this phylogenetic tree that lead to the de-evolution of this particular ability. So if we basically look at the system that these loose jaws have evolved, they produce a brand of light that's rarely visible to other organisms in the deep sea. They have associated with that a set of genes that have evolved to take advantage of that and see red light. Together, that makes them one of the best predators in this particular habitat, and able then to use an ecology of light that is not available to other species in that area.